So this forms the stage 1 of de novo synthesis of cholesterol. That is, first in the first step, what happens? As two molecules of acetyl CoA condenses together to give acetoacetyl CoA with the release of CoASH by thiolase enzyme. The acetoacetyl CoA then condenses one more molecule of acetyl CoA. So a total of 3 molecules of acetyl CoA or 6 carbon molecule is now formed, which is 3-hydroxy 3-methyl gluteryl CoA. So again, one more molecule of CoASH will be released by cytosolic HMG-CoA synthase enzyme. So this reaction happens in all the nucleated cells of the body, provided it is a cytosolic fraction, because only the liver can synthesize HMG-CoA by mitochondrial fraction of HMG-CoA synthase. And then in the mitochondrial HMG-CoA synthase converts acetyl-CoA or acetoacetyl-CoA to HMG-CoA in the mitochondria only for ketone body metabolism. If the liver cells are responsible for synthesizing cholesterol molecule from the same acetyl-CoA molecule, then it is not the mitochondrial reaction that is happening. The same reactions will be simultaneously happening out in the cytoplasm also. So mitochondrial ketone body metabolism will be initiated with the same compounds, with the same enzyme, but the mitochondrial fraction. In the same liver cells, but in the cytosol or liver parenchymal cells carries out the same 3 steps, the two steps of this reaction by the cytosolic fraction to synthesize cholesterol. The mitochondrial simultaneous site, the same reaction may be happening, but there the purpose is a ketone body formation. But in the cytoplasm, the same reaction is happening for the de novo synthesis of cholesterol. So that's a basic difference. So the reaction is it's the same that is happening in the case of KB synthesis, ketone body synthesis, and also in this cholesterol biosynthesis. But the basic difference is that this is cytoplasm and the other one is mitochondrial. Okay, so HMG-CoA is formed, and once this 6-carbon HMG-CoA is formed, it undergoes a reduction by HMG-CoA reductase enzyme, and this is a rate limiting enzyme of the pathway, and it is a reductive biosynthetic reaction, hence uses 2 molecules of NADPH and reduces the HMG-CoA to give rise to mevalonate. I'll show you the structure. So with this, the stage 1 is over. So out of 5 stages, the first step is the formation of mevalonate, and initiated with 2 molecules of acetyl-CoA to give rise to 4-carbon acetoacetyl-CoA by cytoplasmic thiolase enzyme with the removal of 1 molecule of CoASH. Acetoacetyl-CoA, which is a 4-carbon compound, adds 1 more molecule of acetyl-CoA to give rise to 6-carbon compound with the release of another molecule of CoASH in presence of cytosolic HMG-CoA synthase enzyme, giving rise to HMG-CoA or 3-hydroxy 3-methyl or beta-hydroxy beta-methyl gluteryl-CoA or HMG-CoA, but in the cytoplasm. And this cytosolic HMG-CoA is now acted upon by HMG-CoA reductase enzyme to give rise to mevalonate, in which the reduction of the HMG-CoA happens with the molecules of 2 NADPH, giving rise to 2 NADP and CoASH. CoASH is also removed because now we are having mevalonic acid. Cholesterol is not a CoA. Cholesterol CoA it is simply cholesterol. So now with the conversion of HMG-CoA to mevalonate, all the CoASH molecules are removed. Okay, now mevalonic acid or mevalonate, which is a 6-carbon compound, is now formed, and this is a rate limiting step. And this is a rate limiting enzyme. So this enzyme decides whether the HMG-CoA has to be synthesized or has to be converted into mevalonate. Right. So I'll go into the I'll show you the reactions. Yeah, this is our acetyl-CoA. Acetyl-CoA, 2 molecules of acetyl-CoA. The 2 molecules of acetyl-CoA with 1 molecule of CoASH is released. Thiolase comes. It joins the 2 molecules of acetyl-CoA to give rise to 4-carbon. The 1, 2, 3, 4. 4-carbon acetoacetyl-CoA. This acetoacetyl-CoA adds 1 more molecule of acetyl-CoA. With this, CoASH is removed by HMG-CoA synthase enzyme. And it's a synthase enzyme, not the synthetase. Okay, if it is a synthetase, then it might have required ATP. So it's not an ATP requiring process, hence it is not synthetase, it is a synthase enzyme. Right? So beta-hydroxy, beta-methyl gluteryl-CoA, the beta-hydroxy beta-methyl gluteryl-CoA. Okay. Now HMG-CoA is acted upon by reductase enzyme, HMG-CoA reductase enzyme, where what is happening? This CO molecule, this CO is removing CoASH. CoASH is removed. So CoASH H is removed, right? So one molecule of two molecules of NADPH comes, and one atom of hydrogen is going with this. So CoA SH is removed. So the remaining three hydrogen atoms, it reduces the C double bond O to CH2OH, right? So this is the mevalonic acid. So actually this carbon atom is undergoing reduction with the release of coenzyme A to give rise to CoASH. So one H ഇവിടെ പോയി. Then remaining two NADPH gives two molecules of hydrogen, that is four atoms of hydrogen. So one hydrogen is released with this CoASH. Now we have got three atoms of hydrogen. So this is the one hydrogen atom. Then these are the remaining two hydrogen atoms. Okay, so all the hydrogen atoms are completely occupied. So one goes along with CoA to give rise to CoASH, and the three hydrogen atoms will be equally distributed. So that C double bond O is now reduced to CH2OH. So CH2OH, CH2, beta hydroxy, beta methyl, mevalonic acid, right? So this is the stage one. Now this mevalonate, what is happening? This mevalonate is now undergoing decarboxylation to give rise to isopentenyl pyrophosphate, right? That forms the second stage, right? So reactions of the second stage, mevalonate undergoes three successive phosphorylation to give rise to mevalonate 3-phosphopyrophosphate, which is converted into two types of isoprenoid units, that is isopentenyl pyrophosphate and 3,3-dimethyl allyl pyrophosphate. See, this is our mevalonic acid, right? That is CH2OH, CH2COH, CH3, CH2, COOH. Okay, so before converting the mevalonate to IPP, the product is isopentenyl pyrophosphate. So now this mevalonic acid is occupied with, or the final product is having phosphate groups, right? So what is to be done? Phosphate group has to be attached. I'll show you this slide. Yeah, mevalonate. So mevalonate undergoes phosphorylation reactions followed by decarboxylation. Decarboxylation followed by phosphorylation. Okay, so phosphorylation is carried out by kinases, right? So kinase enzyme comes. So wherever kinase comes, there we will be having ATP. So three molecules of phosphate are getting attached. So what happens? Three molecules of ATP also comes. So wherever ATP is there, magnesium also should be there, right? So because ATP can function only in presence of the cofactor magnesium. So kinase comes, wherever kinase is there, there also ATP will be there. Wherever ATP is consumed, magnesium should be there, because the reaction can happen only in presence of magnesium. So three molecules of phosphate is getting attached. So three ATPs will be coming, and what is happening? Decarboxylation takes place. So mevalonate undergoes decarboxylation. So mevalonate will having 6 carbon atoms. So when decarboxylation occurs, what happens? The 6 carbon is converted into a 5 carbon compound, and the 5 carbon compound which is phosphorylated, which is known as isopentenyl pyrophosphate, right? And there is a second molecule of mevalonate also comes, which also give rise to isopentenyl pyrophosphate, but one of the molecules of the isopentenyl pyrophosphate IPP will be immediately isomerized to give DPP, dimethyl allyl pyrophosphate, because for the reaction to proceed, we require one molecule of IPP and one molecule of DPP. So out of the two molecules of IPP formed, one of them will be undergoing isomerism to give DPP. Now I'll show you the structure, right? The mevalonate undergoes kinase. So the kinase phosphorylations are happening in a stepwise manner. The three ATP do not come in a single step. So first mevalonate kinase consumes one molecule of ATP to attach the first molecule of phosphate, giving rise to mevalonate 5-phosphate. See, here there is no the decarboxylation happens only after phosphorylation, right? Now the second step, the second phosphate comes. So the compound will be, or the enzyme will be named as mevalonate 5-phosphate kinase, or phosphomevalonate kinase. So the second molecule of phosphate also will be attached. In the third step, the third phosphate is attached, but there the phosphate is attached onto this OH. Okay, we were in the beta position, or the third position we were having an OH. So the H is replaced with the third phosphate group. So which is known as, this is mevalonate 5-pyrophosphate. So it is known as pyrophosphomevalonate kinase, converting the mevalonate 5-pyrophosphate to mevalonate 3-phospho 5-pyrophosphate. Okay, now it is undergoing decarboxylation to give rise to isopentenyl pyrophosphate. So the here the carbon dioxide will be removed. Right, so from the terminal position, the carbon dioxide is removed. So this bond jumps to this position. So CH2 double bond C, CH3OP, CH2, CH2OPP. That is this. CH2 double bond C, CH3, and this inorganic phosphate also will be removed. So see, because a double bond and phosphate can never exist on a single carbon atom. So the pyrophosphate, the inorganic phosphate also will be removed along with this carbon dioxide. So CH2 double bond C, CH3, CH2, CH2, OPP. This is known as isopentenyl, isopentene, the 1, 2, 3, 4, 5. So isopentane, I know, since it is having a double bond, so compound is known as isopentenyl pyrophosphate. Now this IPP undergoes one of the IPP, two molecules of IPP will be formed because for the further steps to be carried out, we require two molecules of IPPs. So one of the IPPs thus formed will be converted into its isomeric form, in which nothing is happening. The double bond is getting shifted from the third and the fourth position to second and the third position. Okay, so this double bond will be jumping to this position. So the compound will be named as dimethyl allyl. Allyl means the same, the methyl group is located, the two side chains are located on the same carbon atom. So അതിനെ indicate ചെയ്യാനാണ് നമ്മൾ allyl എന്ന് പറയുക. Okay, so here ഈ കാർബണിനോട് തന്നെയാണ് രണ്ട് methyl groups attached ആയിട്ടുള്ളത്. So it is known as dimethyl allyl pyrophosphate. Dimethyl allyl pyrophosphate 3 3 1 2 3. So 3 3 dimethyl allyl pyrophosphate. So that forms the second stage, right? Formation of IPP and GPP. So now we are having one molecule of IPP and one molecule of GPP. See, mevalonate undergoes three stages of phosphorylation reactions. First, 2-phosphate is attached onto the terminal position to give rise to pyrophosphate, while the third phosphate is attached onto the beta hydroxyl group. The HOH is replaced with OP, followed by decarboxylation reaction to give rise to IPP. And there is one more mevalonate molecule giving rise to the second molecule of IPP, and the second molecule of IPP undergoes immediate isomerism to give rise to DPP, in which the double bond is getting shifted. Now what happens? This IPP and DPP joins together to give rise to the 10 carbon compound, what? Geranyl. It is not genanyl, it is geranyl pyrophosphate. It's not N, it's R. Okay, geranyl pyrophosphate. Simple, nothing. What is happening? The IPP and DPP joins together. Or inorganic pyrophosphate അങ്ങ് പോകും. Because ഇവിടെ total ഇപ്പൊ നമുക്ക് നാല് phosphate വരും അല്ലേ? Here 2 phosphate are there. Here also 2 phosphates are there. So when these two joins together, what the net effect is that they will be having 4 phosphate. It's not possible. So 2 phosphate will be removed, and the remaining compound will be having also again 2 pyrophosphates or 2 phosphate, which is known as pyrophosphate. The compound is known as geranyl pyrophosphate. It's a 10 carbon compound, and this geranyl pyrophosphate adds one more molecule of IPP with the removal of again 2 phosphate groups to give rise to farnesyl pyrophosphate. I'll show you the reaction. See, this is isopentenyl pyrophosphate. So one IPP and one DPP acted upon by transferase enzyme to give geranyl pyrophosphate. See, the double bonds are in an isoprenoid side chain in an alternative positions. Okay, to give rise to the 10 carbon, the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right? So 10 carbon geranyl pyrophosphate acted upon by another transferase enzyme which transfers one more molecule of IPP to give rise to 15 carbon farnesyl pyrophosphate. This farnesyl pyrophosphate now adds one more molecule of FPP, farnesyl pyrophosphate. This is GPP. So this is IPP, this is DPP, this is GPP, this is FPP. So 2 molecules of 15 carbon FPP is joined together to give rise to squalene. So again, it is also a NADP is requiring enzyme reaction. The enzyme is squalene synthase. giving rise to squalene.